Om Swastiastu, good morning. My name is Jankarta Sutija with the last three-digit NIM 080, and I'm going to present our student project entitled How We Develop Phytochemical Screening by SGD Binam. Starting off with Chapter 1, Introduction. So the exploration of plant-derived compounds known as phytochemicals has evolved into the cornerstone of medicinal research, offering a treasure trove of bioactive molecules with diverse therapeutic potential. While invaluable, often these traditional knowledge of healing properties of plants lack the precision and systematic rigor required by modern scientific inquiry. The significance of phytochemical screening lies in its ability to bridge this gap, providing a structured and evidence-based approach to the identification, isolation, and characterization of bioactive compounds. So here are the problem formulations that we have formulated. First up is what defines phytochemicals. How can we categorize phytochemicals systematically? What roles do phytochemicals play in medicine? To what extent are the effectiveness and safety of phytochemicals understood? How are the phytochemicals systematically screened? What principles guide phytochemical screening? What is the methodology for developing phytochemical screening? And lastly, what challenges are associated with phytochemical screening?
Good morning, teachers and friends. My name is Anastasia Andretan-Berius, and my last three-digit numbers are 154. This is the objective of our student project: to understand the classes and diversity of phytochemicals, the roles of phytochemicals in modern medicine, the efficacy and safety of phytochemicals, the application of phytochemical screening, the principles of phytochemical screening, the methodology for phytochemical screening, and the challenges in phytochemical screening. This is the benefit. Theoretically, the student project can expand the writer's knowledge, specifically regarding the topic of developing phytochemical screening, including a comprehensive understanding of the classes and diversity, roles in modern medicine, efficacy and safety, application, principles, methodology, and challenges of phytochemical screening. And practical benefits for writers. It is expected that this student project can be an opportunity for the writers to practice and enhance their process in writing a literature review and analyzing the information relevant to the topic of the paper. It is also hoped that this student project will allow the writers to gain more knowledge and a deeper understanding of the medical world and further enhance their medical skills. And for readers, it is much hoped that the student project can provide useful information for the community regarding the topic of developing phytochemical screening and give the members of the community a deeper understanding of the topic. Thank
Hello, my name is Gede Kanjaya with last three digits of my student number is 139. So I'll explain you about the classes and diversity of phytochemicals. So number one, there is terpenoids. So terpenoids play a significant role in medical fields due to their various pharmacological properties and health benefits. Some of the beneficial effects of terpenoid includes antioxidant effects, anti-inflammatory effects, and overall cardiovascular health effect. And the second is phytosterol. Phytosterol are plant compounds that mimic cholesterol and can help lower LDL cholesterol levels in our bloodstream. Their cholesterol-lowering properties makes them valuable in the management of cardiovascular health, especially for individuals with high cholesterol levels. The next kind of phytochemicals will be explained by my friend. Thank you.
Hello, let me introduce myself. My name is Jenny Sujaya with the last three digits of number 8037. Now let me continue the presentations with the other classes of phytochemicals. So the next one I'm going to explain is polyphenols. Polyphenols are organic compounds synthesized by plants that share the same chemical characteristic with phenolic substances. These compounds are known for their bioactivity potential, such as the ability of oxidative on inflammatory stress regulations, helps with micronutrient digestions, and shows probiotic-like effects on gut microbiota. And in plants, polyphenols serves various functions such as attracting pollinators, defending against UV radiations, and protecting the plant from invasions. The next one is alkaloid. So alkaloid is categorized into nitrogen-containing compound, and beside the nitrogen component, there are also other components like carbon, hydrogen, and also oxygen. Alkaloids can be usually found in plants with flowers. And the last one is organosulfur. Organosulfur compounds are secondary metabolite which has certain biological activities and has a unique strong smells that characterize the allium species. The organosulfur is synthesized in plants to serve two main functions, such as protection mechanism against the biotic stresses and as pollination mediators. That's all for me. The next part will be explained by my friend. Thank you.
My name is Ariela Kedisarjo with last digit name 247. Now I will explain about the roles of phytochemicals in modern medicine. Phytochemicals are plant-based bioactive compounds produced by plants. Some of the significant phytochemicals are carotenoids, polyphenols, saponins, dietary fibers, and certain polysaccharides. They are extensively used worldwide for the treatment of various kinds of fatal disease like cancer, neurodegenerative such as Parkinson's and Alzheimer's, cardiovascular disease such as heart attack and stroke, type 2 diabetes, and many others. Phytochemicals are supportive in the prevention and healing of following infections and illness. First, phytochemicals stimulate the body's immune system against disease-causing microorganisms. Second, inhibit carcinogens, which are formed in the body due to the substance we take from the environment in the form of food or beverages. Then third, diminishes oxidation, the damage of cells that arises with aging and exposure to pollution. Free radicals in oxidation may lead to cancer. Fourth, slow the growing rate of cells which can cause cancer. Fifth, reduce tenderness which can cause cancer growth. Six, activate apoptosis process of the injured cell that may be a forerunner to cancer. Seven, not only stop the damage in DNA but also useful in its repair mechanism. And the last one is accommodating in the regulation of hormones like insulin and estrogen in the body. The excess level of these hormones will increase the danger of colon and breast cancer. I guess that's all from me. Thank you.
41, I will be continuing the explanation about the roles of phytochemicals in modern medicine. The first one is phytochemicals as an anti-cancer activity where they can neutralize free radicals in body. They also can inhibit the growth and proliferation of cancer cell by interfering the pathway and promote apoptosis. The second one is anti-inflammatory, or phytochemicals that found in turmeric and quercetin in fruit and vegetables may help modulate the inflammatory process. The third one is as the cardioprotective effects. Phytochemicals that found in beets, hibiscus, and cocoa may help reduce the blood pressure, improve the cholesterol level, and protect the heart. And lastly, the neuroprotective effects, where phytochemicals like flavonoids and polyphenols potentially reduce the risk of neurodegenerative disease such as Alzheimer's and Parkinson's. That is all from me. Thank you.
Hello, I'm Amanda Devasha and my last three digits of student number is 068. Now I will explain about the efficacy and safety of phytochemicals. Phytochemicals are bioactive components that naturally occur in plants and their presence and secretions vary from one plant to another. They play an important role in human health through various effective means such as antioxidant, cell differentiation effects, effects of DNA metabolism, and maintenance of DNA repair. Based on research, the intersection between the predicted health effect of phytochemicals and the ethnopharmacological use of plants containing phytochemicals show that 31% of the predicted health effect have ethnopharmacological evidence. In all the regulations set by the government for phytochemical consumption, the safety, labeling, and health claims should be properly monitored and researched. The in vitro studies have been properly done on the use of phytochemicals, but the in vivo part is still unexploited. There has also been a lack of study on toxicity on humans and has not been clearly demonstrated. That's all from me. Thank you.
Swastiastu, my name is Fiona Aurelia with the last three digits of my student number are 125. In this part, we'll be talking about the application of phytochemical screening. Phytochemical screening is used widely in various fields, including the medical field. It is primarily used to identify and analyze the chemical composition of plant extracts. Here are some of the applications. 1. Identification of plant chemical components. Phytochemical screening will help medical fields by identifying the chemical components of plant extracts. For example, flavonoids, steroids, terpenoids, saponins, and tannins. 2. Evaluate the biological activities. So phytochemical screening is also used on evaluating biological activities of plant extracts. This means we will be able to know what this component may be beneficial for medical fields, such as anti-inflammatory, antimicrobial, analgesic, and antioxidant activities. 3. Counting the quantity of phytochemicals. Phytochemical screening will also be used to quantify the amount of phytochemicals in the extract, for example, the total of flavonoid content and the total of steroid content. 4. Quality control for herbal medicines. As we know, many patients still choose herbal medicines. Thus, there must be a way to make sure that herbal medicines are safe enough for the patient. Phytochemical screening will help on identifying and authenticating herbal medicines, which will detect if there is a presence of contaminants in it. That is all from me. The presentation will be continued by my friend. Thank you.
Good morning, doctor. My name is Joseph Ngesiwe with the NIM 250. Today I'll be describing the principles of phytochemical screening. So the extract can be separated through TLC, which is known as thin layer chromatography. The extracts are added into a glass container coated with silica gel or other absorbent, which will be stored in a chromatographic chamber containing a suitable running solvent. This technique contains two different phases, the stationary phase and the mobile phase, to separate the compounds based on their polarity. The isolation of the bioactive components takes a long time, but it can recognize the known metabolites in the extracts. And that is all from me. Thank you.
Hello, my name is Anak Agung Ayukiran Darmatami, and the last digits of my student number is 133. Today I'll be presenting about the general methodology and methodology for developing phytochemical screening. First is plant material collection. The first step involves the collection of the plant material. This could be leaves, roots, stems, or any other part of the plant that is believed to contain the phytochemicals. There is a selection of plant material, collection and authentication, and drying. Next is extraction. Now this involves a few steps, one of which is selection of solvents, extraction techniques, and optimization. Next is phytochemical screening. So the extracts are subjected to various qualitative tests to identify the presence of different phytochemicals. Some of the important phytochemicals include alkaloids, flavonoids, phenolics, tannins, saponins, steroids, glycosides, and terpenes. Now, there are a few steps to doing phytochemical screening. There is preliminary tests, specific tests, and metabolic approaches. Then the last step is evaluation of pharmacological activity. The extracts are then evaluated for their pharmacological activities. This could include antioxidants, cytotoxic, antimicrobial, analgesic, anti-inflammatory, antidiarrheal, and other activities. And next is further analysis, which will be discussed further by the next presenter. That is it from me. Thank you.
Hello, my name is Keso Timothy Wangdian with 102511110. That's my student number. For further analysis in phytochemical screening, there is isolation and characterization of active compounds. For various techniques such as chromatography and spectroscopy can be used for this purpose. After isolation, the compounds are characterized to determine their exact structure and composition. Next, the determination of mechanism of action. Studies can be conducted to determine how these compounds exert their pharmacological effects. This could involve in vitro and in vivo studies, as well as molecular docking and other computational methods. And also, there is a bioactivity test where the isolated and identified antioxidant components in the plant may undergo further bioactivity tests, which are mediated by free radicals. And lastly, the development of drugs, where the active compound identified in the plant can be used as a starting point for the development of a new therapeutic agent. That is all from me. Thank you.
Hello, doctors. My name is Nafinata Susanta, and the last three digits of my student number is 051. Now I'm going to explain about the challenges in phytochemical screening. Phytochemicals have long been indicated to have a plethora of pharmacological effects. It is important, however, to remember that the current knowledge and stance towards phytochemicals and its screenings is mainly based on in vitro studies. This means that the applicability of phytochemicals in a clinical setting remains as mostly uncharted territory in research. For example, some compounds that have been observed and screened in vitro indicate the presence of antimicrobial effects in some plants. During in vivo, however, these activities have little to no antimicrobial activity. More research is needed to find out about potential synergism and antagonism of certain compounds. The main limitation of phytochemical use is the lack of standardization in related treatments. This is also the cause of low credibility regarding the use of phytochemicals. Clinical trials and screenings evaluating the effectiveness of a certain plant's medical effects are limited. More specific and sensitive bioassay techniques are required to test plant extracts and essential oils. Another challenge is that plant extracts may vary based on environmental conditions and geographical area. These factors influence the availability of medicinal plants, and plants grown in different environments have to be screened and tested individually due to their different components and medical qualities. These challenges can be tackled as the field of biotechnology face advancements which can allow further screening and research regarding the compounds of phytochemicals and allow the development of more sophisticated techniques to identify various bioactive compounds and their chemical structures. That is all from me. Thank you.
My name Pandey Nyomanima Pratista, relation digit of my student number 201. Now I would like to summarize our student project. Terpenoid, polysterol, polyphenol, alkaloid, and organosulfur compound were emphasized as the main class that has significant impact. Photochemical screening involves qualitative tests to identify compounds like alkaloid, flavonoid, polyphenol, and more. In photochemical screening, plant extract-rich secondary metabolites are prepared from the various parts, and standard tests, including the thin layer chromatography, are employed to analyze the component. The method, while economically viable, has low efficacy due to prolonged isolation time and low sensitivity and selectivity. Preliminary tests including high throughput screening and biosensor technology, rapid identify major petrochemical class. Specific tests employ advanced chromatography technique like DCMS or gas chromatography mass spectrophotometry for precise compound identification. And then advice for this student project, the methodology of developing photochemical screening has evolved significant incorporating modern technology in interdisciplinary approach. Thank you for your attention. This is presentation from SGDB 6. Thank you.